choose public. But it might also have been a problem on my side because later I saw there were some comments on, so I'm not sure. But on my page it says that it was non-listed live, and because of this it cannot.
I get the impression that we are live, right?
Yes, we are. I
Okay,
already got the message, and we have the little thing on the top.
Thank you, guys. Thanks. So let's see, it's 11. We'll give our colleagues another minute to join us, and then we'll get started. Welcome you all. Well, good morning, afternoon, evening, whatever it is that you are. Welcome back to the fine seminar. Before introducing our speaker today, I want to thank Julia Osner. Let me just, since I have this ready, I'm sharing the screen. So last week, we heard from Julia about her project in Thailand on macaques, on the long reach of seasonal reproduction in wild macaques. Next week, we're going to have a presentation by Sonia Weil, who is a postdoctoral fellow at the Max Planck Institute in the study of animal behavior, and she'll be discussing cognition and social learning in birds and mammals. We will repeat this next week, but in case it helps you, we unfortunately, the talks in May 14th and May 21st, our colleagues had difficulties. I mean, and they have been canceled. So we will not have fine seminars on May 14th and May 21st. Sonia's next week is going to be the last one for the semester. And again, we'll send you an announcement, but you can mark your calendars. We're getting back on September 3rd. We're trying to plan something special to celebrate the 4 years of our fine community. And so today we're going to hear from Amiyaal Ilany. Amiyaal, let me stop sharing this so I can see your faces. So Amiyaal studied training in biology and computer science. It's always good to understand how our colleagues get to do all the math and computer science. You start early. So his first degree in the University of Tel Aviv was both in biology and computer science. He went on to do his master's and his PhD also at the University of Tel Aviv in zoology. Following that, he came to the States. First, again, further developing his mathematical and computer skills, he did a postdoctoral fellowship at the University of Tennessee in Knoxville at the National Center for Mathematical and Biological Synthesis. After that, he went to Penn. I missed him for a year. I mean, he was at Penn doing a postdoctoral fellowship in biology. In 2015, returned to Israel, where first he was a lecturer in biological sciences at the University of Bar-Ilan, where he's now, and since 2022, he's an associate professor there. His focus has all these years been on studying social evolution, and one of the things that he's spent quite some time, and it shows in his publication record and in the grants he's obtained, is thinking hard and thoroughly about what we can get out of the social network analytical framework. And so I know that he will be doing some of that today. Something else that I noticed from browsing his CV, he has done an unusual amount of outreach. And from our conversation yesterday, I could tell that he really likes doing that. He must be very good at doing it. So it's another way in which we let a broader community of citizens learn about all we're doing as behavioral ecologists and evolution biologists. So with that, I welcome you all, and I welcome Amiyaal. I want to thank him. I want to say that he's made a huge effort. He was, not to mention the difficulties that our colleagues and friends and maybe families in Israel are going through, but he was also not feeling that well the last few days, but here he is. I thank you so much, Amiyaal, for volunteering your time, and please take it from here. It's all yours.
Thank you very much, Eduardo, for this kind introduction, and I'm really happy to be able to present my work in this seminar. I really like this community. I must admit that following COVID, I had this Zoom fatigue and preferred to go to real seminars. But when I discovered this series, I do try to join. The time for me is sometimes challenging, but then I try to at least watch it later on YouTube. With that, I will share my presentation. So I changed the title a bit. Eduardo mentioned that there was no fine seminar on hyraxes, so I promise there will be a lot of hyrax slides. But the theme will be about animal sociality quantified in terms of interactions, a bit of transmission, and social networks. And let's start with an example of the value of sociality. A video taken by my current PhD student, Pramik Kannan. You can see a fox chasing a hyrax. I think at this stage, the fox realizes the hyrax is actually too big. They usually run after pups. And then the hyrax gets some help. It was a juvenile. And with help from a bigger hyrax, they now chase the fox away. Okay, so in this forum, it is maybe trivial to ask the question of why study sociality. I spend more time on convincing my audience about its importance. I don't know why my slides are jumping some of the time. Is there anything about timing here? I hope they won't.
But because it happened yesterday, right?
Yeah, I
And we
think
forgot to check. Maybe you have
like
it on automatic.
timing, but then maybe it's kind of stayed somehow. Anyway, I hope it will be okay. I will go briefly on some examples of the importance of social structure. And it is important to remember that a lot of animals spend most of their time alone. A lot of species are solitary. So being social is not trivial. And even within social species, there is so much variation in group sizes and in structures. So the first important effect of sociality is on longevity. This is a study on humans, a meta-analysis that showed that when summarizing about 150 studies, some measures of social relationships, their effect on longevity is as important as the effect of smoking, which of course in the opposite direction, and more important than the effect of alcohol consumption. I need to find a way to stop this automatic jumping.
Take a minute. If you need to stop sharing, I agree. I mean,
Yeah, let's take a second to do that. Because it will.
I think you can check the presenter view, Amiyaal, and then
I'm not
for some
using
reason
presenter view, so
okay.
But I think I just did a rehearse yesterday, and then it saved the timings for some reason, and that is maybe just try play from start.
But I think when you click on the presenter tab, there is a box that says "Do not use the rehearsal time."
Okay. Is it like at the bottom?
No, it should be on the top. and then you have like in the menu options for the presenter option tab. Then you click on it, and then you'll show like you can have presenter view or notes or something, and then there should be a box there.
Don't have a presenter. I have like slideshow, and that's where there is one
Yes,
option
it's
to
under slideshow.
Okay. Oh, here it is. Here it is. I changed it to manually, and now we can go back. Okay, do you see the slides again?
Yep, we're okay.
Okay,
Yeah.
I hope it's solved, and sorry for this. So I was saying that longevity was found to be more important than several factors that we know that affect longevity, including alcohol consumption, physical activity, BMI, air pollution. Social relationships seem to be more important than most of these effects. And in recent years, some studies in animals showed some parallel results, and I'll talk in a bit more detail about results from our hyraxes. A second importance is of reproductive success, and in these cases, it is usually the females who invest more in raising their young, and it is often found that females that are better integrated socially are more successful in that. A third example is of disease transmission, and here is a nice example where an ant colony was tracked using those barcodes, so interactions were recorded automatically, and then in an experiment, the colony was infected with some fungi, some of the ants those with black circles, and the researchers saw social isolation trying to prevent full infection of the ant colony, resulting in some changes in the social structure compared to the pre-treatment stage. For example, is a recent study showing effects of microbial transmission depending on social factors. And here we see the gut microbiome on the left and the oral microbiome on the right. So we see that strain sharing rate, or basically microbial similarity, is the strongest first within the same individual in different samples, but then in mother-offspring pairs and within household and with adult twins. And looking at oral microbiome, it's again mother-offspring that is strong, father-offspring, and also partners. So whenever you are kissing your partner, think about the interesting microbes that you transfer to each other. And a fifth and last example is the importance of social structure to cultural transmission. So I always like to present this nice example led by Lucy Aplin, where they showed that they can initiate a simple culture in great teats, with some individuals preferring opening a box using the blue side and some the red side, and individuals basically copying each other depending on their social associations. So because of all of these important consequences, I think it is important to understand the causes of social structure, how basically networks form and change over time. And it has become kind of my niche to be focused on that aspect of social networks. And the main theme here, with some diversions, will be what factors determine the structure of animal social networks. And the thinking about social structure is not new at all. This is a slide from a paper by Hind in the 1970s. showing interactions as a basis that can be summarized by relationships between individuals, and these relationships can be summarized by structure, or what I think we call today as networks. And all of these levels may be affected by different effects, such as age, sex classes, blood relationships, status, and so on. But how do we measure, or how do we quantify sociality? I think this is a really challenging part in our field in studying social structure, because it really depends on the species we study. And here is an example from work done by my student Nikola Dragic on Arabian babblers, a social species living in the south of Israel. And Nikola observed them carefully and created these 6 different networks that show interactions based on different behaviors, such as playing, allopreening, proximal foraging, and so on. As you can see, these networks are quite different from each other. And he also found that there is almost no correlation between these networks, which suggests that if we're trying to say, okay, A and B are connected, they are friends, or they have this some strength of relationship, what is it actually based on? What are we measuring? What are we looking? I think this is something that is still requires a lot of work, and a lot of time we just measure something that we can, and maybe are missing measures that we cannot count for some reason. So now some introduction on rock hyraxes, which is the main study, the main species that I study. Hyraxes are part of the weird Afrotheria group of mammals that includes elephants, manatees, and some weird creatures such as sengis. Most of these species live in Africa, but some of them can be found in other areas. So a hyrax may look like a rodent, but it's pretty far from being one. Currently, there are 5 recognized hyrax species. The most common one is the rock hyrax. Procavia capensis can be found all the way from Cape Town. Capensis is the Cape of Good Hope, all the way north in many parts of Africa and some parts of the Middle East. I think maybe the more northernmost point is in Lebanon. And other hyrax species are more reserved to Eastern Africa, including 3 species of tree hyrax, and the bush hyrax, which interestingly can be found in mixed species group with rock hyrax. The bush hyrax is somewhat smaller. And if you see me in the street going with a sad face, that's because I was born 35 million years too late. If I was born in the right time, I could study all these amazing hyrax species, including the titanohyrax and Kvabebi hyrax, species that look more interesting than the little Procavia that we have here at the bottom. They would probably kill me on my first day in the field, but the heyday of the hyraxes is long gone. So we study hyraxes in En-Gedi on the shores of the Dead Sea. This study was started by Koren, who is now a colleague of mine at Bar-Ilan University when she was a master's student, and is now managed together with Eli Geffen, who was the PhD advisor of both Lee and myself. And the location is here in the Judean desert, right near the Dead Sea in the eastern part of Israel. This is how it looks. It is a harsh desert, and Gedi is actually an oasis with several springs, which allow this vegetation and important species of animals to exist. And within this area, hyraxes live in mostly in groups. and they have home ranges that have some overlap. So you can see here some examples. For example, in this area, there are a few big Sisyphus trees, which several groups may visit from time to time. But we see very little interaction. It's very rare to see interactions between hyrax groups. They seem to avoid each other most of the time. Their main predator used to be leopards until about 15 years ago, when the last leopard was gone in the Judean desert, and the leopard is currently locally extinct in Israel. And this is the Arabian leopard, the Panthera pardus nimer, that is extinct in most of its historical range and can be found now only in Oman and hopefully Yemen. And Eduardo mentioned my background in that I started in computer science and biology, but I actually started earlier. This is my father, Amiyaal Ilany, and he studied leopards in the Judean desert, basically in the same location where I study now. So the leopards are gone, but I at least get to do my research at the same location and to study the leopard's main prey, the hyraxes. So currently, the main predator of hyrax adults is the wolf, and this is the main danger to adult. But hyrax pups are can be trapped by several raptors and foxes. So the chance of a hyrax pup to reach its first year is about 10%. So I started my PhD project working on hyrax singing, which I won't talk about here. Hyraxes have these very interesting vocalization patterns, and the males emit these complex songs. And while sitting and observing hyraxes for a lot of time, and we mark them so we can identify each individual, we collected a lot of data on interactions between marked individuals, and I was wondering what can be done with these data. Just to mention, we mark adults with collars. You can barely see the collar pattern here, with tags that cannot be seen in this picture. And young individuals are marked with a unique set of earrings, so we can identify them with from a distance with a telescope and say who it is. And then I heard a talk about networks, and I realized that this can be very interesting to me, and this is maybe the tool to use all of these records, and Koren had a lot of observations of her own to analyze these social patterns, social interactions. And together with Adi Barokas, we looked at effects on longevity, social effects, and did this marker capture analysis and found that basically, or I should say maybe earlier, that hyraxes live in groups, but then females stay in their natal group usually for life. In the rare cases that we observed females moving to another group, it was always from a bigger to a smaller group. And males, upon reaching adulthood, usually at the age of 2 to 3, they disperse, they leave their natal group. They sometimes stay not far from it, but then may go farther away with the years, and they spend most of their time alone. And at some point, a male may reach another group and manage to become the resident male in this group. So the resident, there is one resident male adult in the group that was not born in the group. And interestingly, this resident male is not always dominant. Actually, more often than not, he is not at the first rank in the group. So we found that the probability of hyrax living in a social group to survive another year is close to 80%. And if you are a bachelor males living solitarily, it's a little lower. But we also found that group size had a negative effect on longevity, where hyraxes living in larger group had lower chances of survival. And then we started to construct social networks. And here you see examples of some groups. The circles, the black circles are females, the white ones are males, and the width of the line represent some measure of the time they spend together. And we were interested in linking centrality, and here the size of the circle represents strength centrality. We were interested in linking that to longevity. And at the individual level, we did not find such correlation. So more central individuals did not survive longer. But then we noticed that some groups were more what we call egalitarian than others. So here, for example, the two upper groups, the changes, the differences in centrality are not so large compared to what you see in the lower groups. And we found that individuals that over the years lived in groups with higher centrality standard deviation, so basically in groups that were less egalitarian, in these groups, longevity individuals suffered from reduced survival. So basically the result means that egalitarian groups seem to promote longevity. And this result joins a not so long list of studies connecting sociality with survival. And most of these studies were done on primates, some studies on cetaceans and other species. And you can see our hyraxes here at the top. And usually the effect is positive, with the exception of the yellow-bellied marmot that found a negative effect of network degree and closeness on survival. So back to the levels of sociality of hind, I'll go to discuss the level of interactions to stable relationships. And this effort has been led in recent years by my former student, Camille Borde, now Dr. Camille Borde. And Camille has been using proximity sensors, which are in the box here attached to the collar. So here you can see a good example of the collar. This hyrax is called DB. And these sensors give us a very detailed account of social interactions. Here is an example of the social interactions of a male and a female over 1 month. This is December 2017. So each row here is 24 hours a day, and then the time is from midnight to the next midnight. So what you can see here is that in the first few nights, they did not sleep together, but they did have, or the black ones are interactions that can be short or long. But they did interact a bit during the day. And then most of the following days, they stay together during the night. It's winter, so they get up pretty late, around 9 a.m. Then they go foraging and they interact less during the day. And they end the day quite early, and around 5 or 6 return to the dens for a long night together. So such patterns to me introduce many questions and challenges. I think the question is, or some of the questions are, how hyraxes maintain multiple such relationships? Do they need a lot of memory to manage these relationships? How do they decide when to interact and when not? What are the factors affecting these patterns and the divergence from these patterns at some points? And here is a different view at the proximity data. when summarizing data from 30 different individuals over a period of about 2 months. And here you can see we are looking at a much like higher view of the day-to-day interactions. Each color shows a different group, and the gray ones are those bachelor males. And here we are getting towards the mating season, where those males approach the groups to try to get chances of mating with those females. So you can see that the group structure itself is not changing a lot from day to day at this level of resolution, but those bachelor males introduce a lot of dynamics. Now, another way to summarize the data is to look at some summary over some period. And here we are looking at a daily total interaction duration during the day. And here are examples from 3 different pairs. So you can see that it's hard to predict how much time 2 hyraxes will spend together, even if they have a pretty strong relationship like here. But we can see a stable trend, and this stable trend is found in more than 80% of the pairs. So it looks like most of the relationships are stable, but they can be at different levels of time spent together. And how they manage that, maybe one hint is some evidence for what I call social allocation. What we show here is that if we look at this, these are the interactions of one female and another female here, and we ranked for each day her like best friend for the day, the one that she interacted with the most. This is rank 1, the second best friend, and so on. As you can see, it's very jumpy. It's not consistent. So every day you spend the most time with a different individual. You don't have consistent best friends. And actually, when looking at the fraction of days that a rank was changed, the first rank is the one that is changing the most. So it looks like if I spend a lot of time with Eduardo today, if I am a hyrax, it is very likely that tomorrow I will not spend a lot of time with him. And maybe that will help me to maintain my relationship with some other individuals. And now we ask, can we not just observe the social network, but also manipulate it a bit and change it? And Camille Borde did 2 types of experiments. These are very light experimental manipulations. One is to add heaps of food that we use for the traps as bait anyway. This is a mix of kohlrabi and cabbage. And as you can see, this causes hyrax to aggregate together and also some aggression. But we are not interested in this immediate response, but in what happens to the network during the days that this manipulation exists compared to a pre-treatment stage and a post-treatment stage. The second manipulation we did is we used a drone as a fear experiment. So you could see a hyrax running away from the drone. And again, we're not so impressed by the immediate response, but we ask, does it cause the network to change during the week of the experiment? By the way, we first tried to use playback calls of wolves, but then real wolves came to the canyon and ruined this experiment, and we couldn't control it anymore. So at least the drone is under our control. So there are several ways that we can analyze the data. One way we did it is together with Naoki Masuda, a network scientist, and he developed a method to look at networks and use a clustering algorithm to basically, for example, if we construct a social network for each day during the experiment, his clustering can tell us which networks are more similar to each other, and then say, we can call this group state 1 and this group state 2, and then try to track and follow how the network moves between those states. And what we see is that given some measures of the network, food basically stabilizes the network. So this is the pre-treatment stage, the treatment and the post-treatment. And when looking at different periods of the day, just daytime, nighttime, or the whole time, you can see a clear pattern of a decreased number of states of the network during the manipulation. So food causes the network to be more similar in structure to its position, its state in the previous day. And the network sort of recovers from that to a less stable condition after the treatment ends. With fear, the results were more complex. But what we see in general is that fear destabilizes the network, especially when we're looking at the whole day period. You can see that compared to pre-treatment, there is an increase in the number of states, and also the network does not necessarily recover quickly from this manipulation, but actually sometimes the number of states increase, and the network does not revert back easily to its previous condition. These were a few examples of, I think, detailed analysis that can be done using proximity data. And I think there's a lot more to do with these data. And I think there are also not enough studies trying to manipulate networks. And after trying to do that, I can say why, because it's quite complex, especially in field conditions. There are many things that are challenging. It was pretty challenging to get this result. And as I said, although my main theme is about the structure of the network, maybe I became interested in that mostly because in hyraxes, we didn't really have what to measure in terms of transmission, but we can say that every interaction is a potential for transmission of information. And now we are interested in transmission of microbes. So what happens when these two hyraxes interact? So I recently became interested in microbiome, pretty late in the game, I can say. And just briefly, the microbiome in the last 15 or 20 years has been shown to be very important to many parts of physiology, and maybe more closer to our field, the microbiota gut-brain axis has been implicated to affect social interaction and communication and social immunity and so on. So the microbiome itself is important, but I think we can also learn from it a bit about interactions. And if you ask microbiome experts, which I don't think include me at this stage, they will tell you that the main rule is you are what you eat, at least in terms of the gut microbiome, right? That's how you get microbes into your gut. I want to suggest that you are what you eat, that's true, but it's also you are who you meet, and individuals you interact with are important to what kind of microbes you get. And we started collecting fecal samples from hyraxes. And the first thing you can do is do this. Of course, you cannot see many details here. This is just some graph showing samples in the columns here and a list of a lot of microbes that are found or absent in each of these samples. The point is that if you're looking at this list, you can just say that two microbes were found in two different, or some microbial species was found in two hyraxes or not. but this does not indicate that they actually got this microbe from the same source. But now using shotgun metagenomics, we can actually infer strain-level transmission. So what you see here is a phylogenetic tree of one species of microbe, and this effort is led by Pramik Kannan, who took the video from the first slide. And so this is some Firmicutes species that has no real name. And what you can see here is the similarity in the DNA of these species across multiple samples. And these samples come from individuals, males and females, from different groups of hyraxes. So what we can see here is that when these leaves are very close to each other, the distances, the branches are very close. This shows that the DNA sequences are very similar, and this is validating that samples taken from the same female EK at different dates are actually showing the same strain. But what we can also see is examples where shown by these red arrows here that indicate transmission, basically two different individuals that feature the same strain. Now, this is not necessarily a direct transmission. It could have been transmitted by a third-party individual, but this is evidence for some close encounter between those individuals. And if we sort of summarize these data, we can ask how is the microbiome shaped by the social network? And here you can see a summary of the social affiliation from the proximity data with groups given different colors, and those bachelor males here in the middle with mostly weak interactions, but with multiple groups, so serving as kind of bridges between groups. And you can see here a group at our second study site, the second canyon where we worked, the David Canyon, socially disconnected, at least from our data, from groups in the other canyon. But when looking at the microbial strain sharing network, this network basically summarizes multiple such trees of showing microbial similarity. We see again some pattern of the groups showing transmission within groups, but also more connectivity between groups where those individuals from David are now actually connected to individuals that they were not recorded socializing with from the proximity data. So this caused me to think, and I'll be glad for opinions here maybe in the end of the talk, that can we actually reconstruct some history of the social interactions from such trees of strain transmission? Here is a phylogenetic tree of another Firmicutes species. And in this tree, you can again see some cases of transmission between members of the same group. But also, I was a bit surprised to see the same strain in two females from two different canyons, one from David and one from Orgot. Now, this could be maybe an error or some fluke, but it could also reflect some interaction that we did not observe, or possibly a third party, possibly a bachelor male that maybe was born with this female, had the same strain, and then interacted with another female and transmitted this strain to her at a later stage. And to illustrate this, here is a map of our study site where we work in those two canyons, Arugot here at the bottom left and David here at the upper right. And over the years, we have had about 3 or 4 cases of males that were born, for example, in David, and at some point, they emerged at our Arugot study population after some long process of dispersal. And I always thought about it as an example of dispersal, that this male will never go back to its original location. But from the proximity data, we realized that for some males, this is not true. This guy called AE, we had known it only from interactions in our gut. We observed it only in one canyon, but from the proximity data, we got clear evidence that it made a few visits to the vid. One of these visits was for 1 day, and the next day he was back in Arugot. Just to illustrate, for a hyrax, this distance is about 3 kilometers. It's a bit like me flying to Australia and then flying back on the next day. But those visits and tell us that males kind of see a bigger world. They roam larger distances than we thought, and they can serve as those bridges and transmission agents connecting between populations. Although this is not the main point of this talk, I do want to tell a bit about the interesting patterns of sexual interaction in hyraxes. This was mainly studied by a former master's student, Einat Bar-Ziv, and hyraxes have a very short mating season, occurring only for about 2 weeks in the middle of summer, so it's super convenient for observation. And during these 2 weeks, we actually realized that most females mate perhaps only 1 or 2 days, but on these 1 or 2 days, they may mate multiple males, including possibly the resident male that lives with them all the year, but also those bachelor males that approach them and are more interested in females during the mating season. And what we see here is a network of only sexual interactions, not social, occurring between females in red and males. This is a resident male, these triangles, and a bachelor male, these diamonds. And what you can see here is that this is an interesting game. I call it the playoffs, and females are approached by males. Sometimes males are approached by females. Although females tend to reject males more often, sometimes males also reject females. And what you can see here is that each individual has a different amount of choices. So for example, these 3 females lived with this resident, and at least from our observations, these are the only interactions they had. So they could choose to mate with the resident or do nothing. In contrast, this female, for example, was approached by 4 different males, so she had more options. And what we find is that considering these local options, she preferred males of higher rank. And this does not hold at the population level. You don't find that females choose, in general, higher ranked males, but locally, they go over their options and choose the best option out of what they got. And this, in the end, results in those parentage data. I know this is a bit messy, trying to cram a lot of hyraxes into these graphs, but what you can see here is basically points from individuals to their offspring, pointers. And the main point here is that although there is some connectivity between the David and Arugot population, these are the David hyraxes, and these are the other canyon, the Arugot hyraxes. And at least from our data, there is no parentage connectivity between them. And recently, looking at the parentage data, we also realized that there is multiple paternity, and at least in half of the litters, we see pups coming from 2 and sometimes even 3 different males. And this is surely an underestimate because we don't have data of all the pups in the litter. How are we doing with time? Not so well. So I will try to do it a bit quicker. I call this part how to build a social network. I think I'll skip that a bit and say that when I moved to my first postdoc, I work with the amazing Kay Holkamp, who studies spotted hyenas and collected so many years of data, including their social interactions. And I was interested in social networks, so I did this complex analysis of what drives hyena social dynamics. And there are multiple factors, including environmental effects, individual traits, but also structural effects such as this tendency to close triads, which was actually impossible to run any model without this tendency. And so I've been finding this pattern of clustering where there is a tendency that if A is connected to B and B is connected to C, then A is also connected to C, and avoidance of these open triads, which seem to be less stable. And I was wondering what kind of mechanism can cause this clustering. And one option could be based on what we call social inheritance, where, for example, in mammals, it's trivial. If you are born to a mother, you get some time to spend with her because you need her milk. And then you get time and chance to meet and interact with the affiliates of your mother, and that or gives the option to close those triads. So together with Errol Akçay, we developed a very simple toy model of networks. The idea is that it's not a statistical model, but we run a simulation. We start with a random network, and at each step, we add some newborn. We choose randomly some mother connected to its mother. And then the main point is that the newborn has some probability to connect to the friends of its mother to close those triads, and some other probability to connect to other individuals. For example, maybe it's age mates or some other trait it is interested in. And to keep the network size constant, we kill one individual at the end of the step. And if we run this simulation for only a few steps, we can see that we start from a random network. The red ones are those newborns, and very quickly we get to a network that has some of the features that we observe in real networks, such as clustering, individuals that are solitary, isolated, and variation with some individuals that are weakly connected and others that are more central. So we fitted this model to networks of several species and found that we can basically simulate and find parameter values to have networks, to simulate networks that are similar in structure to the observed networks. But then we asked, does it happen in nature? Do individuals actually inherit the social relationships of their parents? So I closed my own triad and connected Errol Akçay with Kay Holkamp, and we used 27 years of hyena data to ask the question, if we have a mother and offspring in a network, and do their relationships, are the relationships similar to each other? So basically, when we have this matrix of association strength, do we find a correlation between the association strength of the mother with other hyenas and that of the offspring with the same hyenas? So we are looking at these correlation coefficients, and we find that they are much higher in mother-offspring pairs than in other pairs in the clan. And also that it actually depends on the maternal pedigree. This is the maternal pedigree, and if we look at distance at this pedigree, a distance of 1 is mother-offspring, so the similarity is the strongest. Then when you go farther away in the tree, you become less similar in terms of sociality. And we see that although the mother-offspring association index, the mother-offspring bond, weakens over time after the offspring is leaving the den, the mother-offspring similarity in association, basically the social inheritance, remains strong for at least 6 years in female offspring and about 4 years in males, because males start to disintegrate from the clan towards the time that they are dispersing. And we also find what I call a silver spoon effect, that in the first year, you don't really see this pattern. From the second year and on, offspring born to high-ranked mothers are more likely to copy their associations than those born to low-ranked mothers. And my take is like, if you are born to successful parents, not even in terms of sociality, just copy what they do and you'll be fine. If you are born to losers, you should try to change and do something else. And in the end, we also find some association of social inheritance with longevity, suggesting that, again, those born to higher ranked females should stick to them in terms of social contacts. And our model predicts that the survival of those who are born to low-ranked females would actually be higher if their social inheritance is weaker. So to wrap it up, talked about different levels of sociality, interactions, relationships, networks. And I think some pattern that seems to be coming up is the value of stability at all of these levels. And when we think about our life, I think stability is so important. And for several years, I thought about myself as studying network dynamics, and I now think maybe I'm actually studying social homeostasis, where individuals are trying to maintain some levels of sociality close to some set point, and what happens when there is some disturbance and they are not successful in maintaining it. But in comparison to homeostasis of physiological traits, I think that in any social networks, a lot of individuals may be actually not near the set point, and that always introduces dynamics and changes in the network. A lot of individuals are maybe dissatisfied with their situation. But all of these are my thoughts, and I wonder what a hyrax would think about all of that. I'd like to thank many people that were involved in this research and our funding sources. And thank you for listening, and I'll be glad for discussion and questions.
Thank you, Amiyaal. Wonderful talk. Lots and lots of things that I'm sure we will be discussing. So thank you all for listening. And very quickly, since I recognize familiar faces, we know the procedures. Just go ahead and in the chatting window, type a question mark if you would like to ask Amiyaal a question. If you're a student, we give you priority. Amiyaal and I were talking about this yesterday. We want to hear from students. You own it. So please don't be shy. Just ask the question, write it if you want us to translate it, if English is not your first language. So with that, we can stop sharing the screen. Well, actually, since I don't see a question mark, oh yeah, I do. Okay, Karsten, you're first.
Actually, there is a student first.
There's a student first. You're right. Eyal, yes, good. Yes, I like it, Eyal. You were asking questions last week. Welcome back. You go.
Thank you very much. For those who don't know me, I'm currently a student in Bar-Ilan University. My question is regarding the migration of males. You mentioned you saw the hyrax AE migrating from the David colony to Arugot. Do you suppose it's an isolated incident? And if not, what do you suppose may cause it?
First, just to be accurate, it's not migration, it's dispersal, okay, that males do when they're leaving their natal group and start wandering alone. But what I showed in this specific graph was something done after at least the initial dispersal, that this male that we knew that lives in one canyon, Arugot, made these relatively quick visits from one day to maybe a couple of weeks long to the other canyon and returned to where it was spending most of the time. So at least one of these, actually the shortest one, the one-day visit, happened during the mating season. So my guess is easy to say that he wasn't very successful in the mating season in the Arugot Canyon. So he went to another place, which is a huge effort in the middle of summer to travel to the David Canyon. And apparently it didn't go well there as well. So the next day he returned to Arugot. The other visits were in fall and winter. So I have no idea what they are about. But I can say that about these dispersing or these bachelor males, they do not have a constant territory after they left their natal group. They move from time to time. I think they're always trying to sort of get a better position towards the next mating season. And I think what's interesting is that, and perhaps that's true for many animals, right? The mating season is so short, right? So basically all this male, what it needs to do is to survive a whole year until the next summer, where it has 2 weeks with chances to be a parent again, right? So it should all, I think, be thought in the context of that.
Thank you. Now it is your turn, Karsten.
Yes, hello, my name is Carsten Schrade, and I'm a researcher at the CNRS in Strasbourg, France, studying mammalian social evolution and doing fieldwork in South Africa on small mammals. Yes, thanks a lot for a very fascinating talk showing many different aspects how one can use social networks to understand social evolution. And I have to apologize for my question. Maybe you answered it in your talk, because my question for the hyraxes you kind of answered with the hyenas later with this inheritance. At the beginning of your talk, you showed results that the degree of egalitarianism is related to longevity. And in your slide, you had 2 examples of an egalitarian group. This was group I in 2001 and group I in 2007. So it seems to be the same group. And for despotic group S in 2004 and group S in 2007. So my question is this degree of egalitarianism versus being despotic kind of culturally inherited in groups and maintained within these groups? Or is this just a coincidence that in your slide you have twice the same group?
Yeah, that's a very good question. And if I understand your question, it is part of like, are there group characteristics that are affecting these things? And I think it is sort of a coincidence that these were examples from the same group for each type, because groups may have changed their egalitarian level from year to year. But I have to say that unfortunately, we don't study so many groups over the years. It's mostly the same 5 groups since the study started. So we cannot tell much about, group traits and basically every difference you find between groups. The question is it because they live in a slightly different location, they have a slightly different diet, so what is it some group, culture or trait, or just random effect? We cannot tell that because of the small number of groups that we follow. That's why we usually try to go to the individual level. And even this result about egalitarian groups was analyzed at the individual level. For each individual, we basically looked at, say it lived 5 years in the same group, but the members have changed and the group structure has changed. So the measure of egalitarian versus non-egalitarian was taken for each individual for the social environment that that it experienced during its lifetime. Again, because at the group level, we cannot really tell that.
Okay, thank you.
Amiyaal, may I?
Yes, so that you know for future reference, Ronnie, I hope you'll join us. Yes, you can ask. What we usually do is you just type a question mark in the chatting window, and we call people by order in which they are. But since I'm the next one, I'll skip myself and welcome you. I hope you'll
Thank you
join
very
us
much.
for further meetings in the future. Go ahead. If you can please introduce yourself, tell us where you're joining us from and institution, etc.
My name is Roni. I'm working in the Open University of Israel and studying Arabian babblers in Israel for over than 35 years. And thank you for the permission. Amiyaal, thank you very much for the interesting talk. Do you have any idea? You found a negative correlation between group size and longevity. And theoretically, at least, we thought that there is some safety in numbers. I would expect that there would be a positive correlation between group size and longevity. Do you have any idea why is it not the case?
Well, of course, there is safety in numbers and other advantages of living in groups. But there is also, I think, the concept of ideal or optimal group size. And I think it is known that this optimal size is very hard to reach because even theoretically, you can think, let's say that the optimal group size is 10, and now another hyrax joins, and let's say they cannot force them out, then they are now 11. And for all of them, it's still much better than 1, right? Than to leave the group, right? Or even much closer to the optimal than to split the group to 5 and 6, right? So a group can diverge from the optimal level, I think, quite easily. And I think what happens in our hyrax group is that at least most of them include 1 resident male and maybe 4, sometimes even 2 females, but maybe 4 to 5 seems to be like an optimal number. But some groups at some points reached even more than 10 females. And maybe that was detrimental. Maybe that made the competition to be hard and there was not enough food or just space in sleeping dens or brought more parasites. And perhaps that was the reason for this effect. We don't know the real reason, but I think that, more is not necessarily merrier in terms of group size.
Thank you.
So I'm next. Amiyaal, so many questions. So it's very exciting talk. I wanted to ask you if you can share the screen again and go back. It must have been during the first third of your talk. You had what I would describe, I mean, if we're thinking chronobiology like an actogram, where you were showing us the data collected of the interactions between males and females during a 24-hour cycle. And my brain has been trained, I mean, so many years listening to chronobiology. So first, I want to be sure that I understand this. If I understand it correctly, it's fascinating, a fascinating data set. So this is technically proximity data, right?
Right.
So whenever, if we get black, it means that the 2 individuals are within a distance that their receivers are recording the other individual. Am I right?
That's correct. I didn't say it clearly, apparently. That means there are, there is always some level of electric noise, but in this case, it's about 1 meter apart. So it's pretty close proximity. Yeah.
Okay. No, because the moment I saw this, and again, it may be completely meaningless, but I just wanted to mention it to you because your emphasis today was on social factors. and you didn't really discuss much the potential influences of temperature and moonlight. And when I was looking at this, I just noticed some whitish blur. So very quickly, I just checked when you had a full moon and a new moon for this month in 2017. The animals are they, I mean, where are they? Because there's a very clear bimodal pattern. I mean, this activity, like you were saying, is you think it's because they're together during the night, sleeping in barrows, and then they're out during the day. Okay, can you comment a little bit of that and the possibility that their temperature and or moon phase influences on this pattern?
Yeah. So they are surely affected by temperature because hyraxes do not control their body temperature very well. That means that in winter, they have to bask when they reach out of the den, and they need to cuddle together to keep their body temperature. And in summer, a hyrax and temperatures in Engedi can go over 40 Celsius degrees in summer. And a hyrax cannot stay in the sun for very long. They will die out of a heat shock. So that means they have to behaviorally adjust their temperature. And since this is winter, they spend, as you can see, most of the night in long interactions. And this is just these two individuals, but perhaps they, at some point, for example, in this gap here, were adjacent to other individuals not recorded in this specific interaction. And they use it to warm up. And in summer, the pattern that can be actually interesting to add a slide comparing to summer, the pattern is quite different. They wake up at about 5:30 with first light. They are active only for a couple or 3 hours early morning, and then they retreat to dens for most of the day because it's too hot for them to be active. And they would go back and be active in the late afternoon for an even shorter period before nighttime. So the pattern will be more like bimodal with morning and afternoon activity. I don't know enough about the effect of the moon. It's something that I'm not sure we checked, but we generally thought that hyraxes retreat to dens, and I don't think they are very active at night, but we do see, social things happening. It's just hard to say because we cannot observe if these social interactions and those changes during the middle of the night, are these all sort of within room interactions, or maybe even going out and moving to a different den? We don't know enough about that.
But okay, but you've answered my question. So overall, these are not animals that you're going to go out at night and see them moving around. I mean, they're pretty much in the den.
I don't think that they are active. They are rarely active at night because it's too dangerous for them. The night is full of predators.
We can talk later. I want to tell you about and I point you at some literature that some colleagues published. It's amazing that you can manipulate some of them by providing them food, because there's so many, I mean, like you did with the social networks. It's fascinating when we can start manipulating conditions. It opens up a whole new world of questions to ask. I mean, not all of us can go and provide food to the animals with the ease that you were showing us. Quite exciting. Thank you so much.
Yeah, and I want to say that we tried very light manipulations. We didn't want to, people always ask me, why don't you add another hyrax to the group or remove a hyrax or something? Yeah, I know that can be done. For example, removal experiments were done in gray tits. It's easy. They can trap some of the birds and remove them for a weekend, and they've seen very interesting effects on the social structure. We are too afraid to do such things, and that the harm may be something that we cannot control. So we tried effects that would change the network, but would not be a major effect. And I'm pretty confident that our added food and the drone experiments are not things that the hyrax remember a week or two after the experiment ended.
Thank you. Do we have any questions coming from our YouTube viewers or anyone else who has a question? I have some more, but if you want to stop sharing, that's okay. I did write down something else. If nobody has other questions, give me a second, because if I don't write down, I forget what I wanted to ask you.
I would like to ask a question.
Yes. Go ahead.
You mentioned the social inheritance in spotted hyenas. Do you suppose that the existence of pups somehow changes the social standing of the mother?
In hyenas?
In hyenas or rock hyraxes?
Well, it's quite different. I didn't have a lot of time to talk about hyenas, and you had, I think, at least you had several talks about hyenas here, right? And one of them by Yves Davidian just a couple of weeks ago. But in hyenas, the hierarchy is very strict, and it is a matriarchal society. So individuals inherit their social rank. My results were about inheriting social relationships, but it is known for a long time that they inherit their social rank, and then this does not change very often. So I don't think that once you have a newborn, your rank as a mother changes, and if it does, just slightly. In hyraxes, again, I didn't touch on that. The hierarchy within a group, actually, we don't see a lot of aggression at all. So it's hard to basically quantify the hierarchy. And I think it's a pretty shallow hierarchy that actually most of the time there is no, there's not a lot of aggression, or I guess what to fight on. And there is no, like, prey and meat to fight on like in hyenas. And I'm pretty, and all adult females have pups every year in hyraxes. So I don't think that having pups would change the status of the mother.
Thank you very much.
I don't see other questions, so I'll go ahead. I thought I am very interested, even when I don't know a thing about it, that, and given your background in computer science, you started talking about simulating social networks, and I get the impression we're going to see more and more of that. Many people with more expertise than I have on computer science and stats and study design are really suggesting that we try to simulate data before actually conducting the study. So I wanted to ask you, because you clearly have some training on that. If we're gonna, when you were describing to us that you simulated the data to see the extent to which you could produce a social network that would have some resemblance to your actual data. What are some of, and again, I'm asking this, this is pretty very basic question, right? I don't have the background, I'm beginning to read about it. So what are some of the basic assumptions? Because how do we stay away from circularity? We only have data on the data we have. And when you're gonna simulate the data, you have to bring in assumptions that for something like the hyrax, you cannot really, it's like we do not have data outside your system, because your system is the only system that has any solid data on hyraxes. So in this exercise, in this approach you took, how does one get started simulating the data?
Yeah, so this is a very good question, and I should say first that the point of our models with Errol-Akçay was to actually put first the data aside, okay? And because when people started using social networks, this was kind of a cool tool, right? I have data, you have data, you observed some interactions, then we can all construct networks and we can sort of find patterns. And the risk is that, and how do we find these patterns? We find them with some statistics that are actually a bit complex in networks because there is interdependence. The data points are not independent compared to when you just collect some trait of individuals. And that means that first, the field is still suffering from some challenges in finding the right statistical methods to account for this interdependence and so on. But also, when you construct a network and you ask some question, it is, I think there is always the risk that you find some pattern that is a result of this snapshot network that you created. And to give an example, when I take a picture of, let's say, some people in the audience, I might erroneously find that, males tend to sit near other males and females near females, or that individuals with blue shirts tend to sit near individuals with red shirts. All kind of different patterns may emerge from this kind of sometimes random snapshot of a given situation. And that is a risk of coming from a data-driven approach. You may find some patterns that you're asking, even if they actually depend on other processes, okay? And what we try to do is forget about the data and try to think about sort of pure processes. And that is, in a way, a naive approach, because of course many processes affect the networks, but we try to kind of come up with first-degree processes. And this idea of social inheritance is a very basic idea. If I connect to my mother and I have some time and I get the chance to connect with the friends of my mother, then what happens, right? And I didn't show it in my slides here, but what we actually find is that this simple process of social inheritance can already explain some patterns that were found in statistical analysis of networks that were attributed to other processes. Let me give you an example. Some papers suggested that social traits are heritable. Now, I don't say that social traits are not heritable, but I say that it is hard to quantify it. And of course, maybe there are some genes for levels of sociality and so on. And there are some good papers showing that. But what we can show with our model is that if I just copy my mother very accurately, I will get a network position that is actually identical to hers. I will have exactly the same levels of connections that my mother has. And that means that it will seem to be that all of my network traits such as between a centrality or so on, are heritable. But I did not have a gene for between a centrality. I just had, say, a gene to copy my mother with some level of accuracy. So the idea...
Hold it there, but actually, because I think I fully agree with you, but then what we shouldn't be talking about is not even bring the word gene, because the only thing we can say is that the offspring ends up having a similar position as the mother. The process, if I'm following you, what you're saying is that we do not really know the process by which that is done. It could be cultural transmission, horizontal cultural transmission. It can be a number of things. And if I follow you, what you're saying is, hold on a minute. I mean, we need not invoke genetic inheritance to explain what you're getting with your modeling. It can be any number of processes that generate this pattern. Am I following? Is that what you're saying? And I agree.
I think yes. And I will give another example. there are also many studies showing assortativity, or the tendency to connect to similar individuals, okay? Now, I do believe that assortativity exists. For example, in the hyena data, we see that age mates are more likely to be connected. But in our model, we also simulated some trait. Let's say there is some trait with a value between 0 and 1, and you inherit it from your parents with some mutation rate, just a simple trait. It doesn't matter what it is, the trait itself. But then if you connect to again, the friends of your mother, if you use social inheritance, then you end up connected to individuals with this similar trait, or at least a similar level, not necessarily exactly the same trait value. So you get those neighbors of, say, yellow individuals and green individuals and so on, but it is not because they want to connect with other yellow individuals. Right. And this is, again, an example of this very low-level process that may explain some other process. And because so many processes are involved, it is hard to reach, it is easy to reach, I think, wrong conclusions regarding social preferences. And I think people need to be very, very careful about that. And if you only look at a snapshot of the network, you will see, I can show you a network simulated with these trait values that shows assortativity patterns. But in this model, because I program it, so I know there is no tendency to connect to individuals with a similar trait value. So the point of our modeling effort is to try to think about simple factors and what kind of networks you get from these preferences. In our case, we started with this pattern, with this process of social inheritance. And the sort of problem is that even with our simple model of 2 parameters, you can already get very complex networks, right? Mm-hmm. So let's say you add now another preference for assortativity, or so for some specific trait value or so on. You very easily get this huge soup of results, which is hard to untangle what are the real factors behind what you observe, right? And this soup is what we observe in nature. We get the end result, and it is hard to disentangle what is behind these results.
I don't know if I'm finding this pattern. Please interrupt me. I mean, it's just a few of us, and nobody has asked, nobody has indicated that you want to ask a question. So let's just open it up. Should we? Well, I mean, do we have people watching in YouTube?
No, I don't think so.
Well,
I think there's only 2 people. One is me. I'm not sure who's the other one.
Would you agree, Karsten? Maybe we can finish our live session and keep talking here. So thanks. If you're watching via YouTube, you're more than welcome. Remember, you can join us if you want to chat a little bit longer, that we may be here. Just join the Zoom. Otherwise, to our YouTube viewers, we look forward to seeing you next week. So I'll stop the live streaming now. I stop the live streaming.